I'm going to provide a relatively brief introduction to neuroimaging. We will have some videos or links to videos that I will not play. If you're interested in them, then you can click on the links and visit those and watch those videos, and I will provide these slides so you can do that. There are two broad types of neuroimaging. We have what we call structural methods and functional methods. So structural methods are the static picture of a brain. Functional methods are looking at some sort of functioning of the brain. So structural methods include computed tomography, magnetic resonance imaging, and then a subset of MRI we call diffusion weighted imaging. And then we also have our functional methods like our functional MRI, positron emission tomography, or the electroencephalography. So today I'm going to provide an overview of some of these methods. What I recommend is watching this video will provide a good introduction to MRI, and it is very brief. But now an overview of common sequences. So currently whenever a patient receives a brain MRI, the neuroradiologist orders at a minimum a T1, a T2, and what we call a FLAIR scan. We'll typically also order a diffusion weighted image. And depending on the quality of the scans and the scanner itself, and really what's needed for the question, the scan would take anywhere from about 20 minutes to an hour. More complex studies add sequences, different types of pictures, higher resolution images, or very specific planes or views of parts of the brain. And I'll provide examples of each of these sequences. So the structural imaging will give us information about the brain at a single time point. We can use this over time, meaning we could do an MRI of someone's brain, 6 months later, repeat the MRI and look for changes. But again, each structural image is just a snapshot. It will provide us some information about macrostructure versus microstructure, and I'll have some examples of that. So macrostructure, brain volumes, gray matter, white matter, the size of the ventricles, which are the CSF, the cerebral spinal fluid filled spaces within the brain. We can see regional volumes. We could measure the global volume of the brain. We can look at the volume of parts of the brain. We could look for vascular changes. That could be a stroke. It could be some other sort of lesion in the brain. We can look at tumors and all sorts of other pathology. So macrostructure are larger features of the brain. So here's an example of macrostructure and how we look at it. When we acquire an image, we will take it in one plane. 3 primary planes: sagittal, axial, and coronal. And these can have multiple names. Axial is also called horizontal or transaxial. But these are the 3 primary planes. So typically for imaging, we acquire the picture of the brain in one of these planes. So it could be sagittal. And then what we can do, we can reslice it so we can look at it in different planes, different orientations. And a lot of the research images that we're going to use in this class are all, they would all look the same in any of these views, meaning they're all equal. The resolution, we'll get to that, what that means, but it's all the same. But sagittal, axial, and coronal are our planes of view. And that's what allows the neuroradiologist or a researcher or someone else to look at the brain. So MRI is really, it's run, what it sounds like. This is magnetic resonance imaging, so it's using strong magnets, this electromagnet. So the MRI is a cylindrical tube, and that houses this really strong electromagnet. The magnetic field inside the scanner affects the magnetic nuclei of atoms, and this is a repeat of that video. Normally, atomic nuclei are randomly oriented, but under the influence of a magnetic field, the nuclei become aligned with the direction of the field. The stronger the field, the greater the degree of alignment, and even the number of, or the percent of protons, these nuclei, that are aligning with the field. Not all the protons and all the nuclei will not line up in the field necessarily, just most of them. When pointing in the same direction, the tiny magnetic signals from individual nuclei add up coherently, resulting in a signal that is large enough to measure. The key to MRI is that the signal from hydrogen nuclei varies in strength depending on the surroundings, meaning where is that hydrogen nuclei, what type of tissue is it? And so this can provide a means of discriminating between gray matter, white matter, and cerebrospinal fluid in structural images of the brain. So here's an example of what happens. Random orientation of protons. Someone goes into the scanner, the protons start to align, and then we use what we call a coil, but use a radio frequency pulse, a signal, to knock those protons out of alignment, and then can measure how they get back into alignment. And so this is a process. This is not a physics class, so we won't get into all the specifics. Don't worry about understanding all of this at this level. That's not what the point of this class is. But we can, as I said, we can use the different properties of protons, this alignment, in different parts of the brain and the body to get an understanding of different types of tissue or whatever's surrounding the proton. So here's an example. So we can use that information to acquire what we call a T1 sequence. And this is the basic structural scan. It provides clear delineations of gray matter, white matter, and cerebrospinal fluid. And so this is really the scan sequence acquired for nearly every clinical and research MRI. Just off the bat, get a T1. So again, we have our different orientations, the slices, a coronal slice, axial, and a sagittal. And then we also use some directions, what we call, so we can get oriented in the brain. So toward the front, so here would be nose, and the eyes would sit right there. And so toward the front of the brain is anterior, posterior, we have superior to the top, inferior to the bottom, and then left and right on these images. And one thing about a lot of images, most structural images are presented in what we call radiologic orientation. So the right side of the image is really the left side of the brain. So it's flipped. And one way to remember that is it's like if you were the physician, somebody approaching the bed. So someone is lying in the scanner and you're approaching them from the bottom, you're looking at their feet. So really what you're seeing is you're seeing that person from underneath. And so everything would be flipped around. So that's one way to think about it. But that's generally the case for most of the imaging we deal with, is the right side is going to be the left side of the brain. And if we're doing a 3D view, then that's not the case. Right is right, left is left. And then often with a lot of functional imaging, then that has been changed. But again, remember, most of the time we're looking at an image like this. What we think is right is really the left side of the brain. So here's a T1 image. So this is acquired for research purposes, the sagittal view of the brain. And I know it's going kind of quickly, but what I'm showing you is there is something abnormal about this scan. And so did we see anything abnormal? Let's go through it again, and then I'll pause when we get to the abnormality. And this really might not know if you don't know anatomy. So right there, and I'll go back a little bit. So right here on the brain, this is really outside the brain, this is in the meninges, there is this area. And so this is the meninges. This surrounds the brain. It's a protective layer, a sac, if you will, around the brain. And so right here, this is an abnormality, but in this case is something benign. It's a calcified area in the meninges. It doesn't put any pressure on the brain. That's obvious. It shouldn't present any clinical symptoms, but it is an abnormal feature. And this could be relatively common, but meaning if we scanned 100 people, we might see it in 1 or 2 or 3 people, which might not seem like a lot, but at a population level, they can be relatively common. And so this is just an example of T1 that we acquired for research, but it does have clinical implications because this is somebody who might not have had a brain scan before, and we look at it and we see this feature, and what we do in that case is a whole other discussion that we won't cover in here, but something to keep in mind whenever we're doing clinically relevant imaging for research. There can be reasons that we need to bring this up. And so in this case, we actually referred the individual to a neurosurgeon so they could get a clinical scan and at least get followed, make sure that it really is benign. Because some cases, it could turn out to be a tumor or some other abnormality. So that is a T1 scan, again, a basic structure of the brain. This is the closest type of scan to what we would see if we could remove the brain. So from this, from these T1 images, we can understand the structure of the brain and how structure relates to behavior. So this is a false color image. It's really, the colors are different gyri, different parts of the brain that you would learn in a neuroanatomy class. This is an example, though, of software that we will use in this class. That you would be able to take a scan like this one and be able to get a 3D model that would be on the computer, and we can use this for research. It's some utility within clinical settings, but typically research at this point. So this is the outer layer of the brain. This is cortex containing cell bodies of neurons. We can use the T1 image to look at structures within the brain. So we can visualize and measure subcortical structures. This blue area, caudate nucleus, green is thalamus, this pink structure is the putamen, and then we can get volume. So again, these are coming from software that we will use this semester to generate these images. So the T1 scan will provide a lot of information. Structure of the brain, we can get volumes and thickness and all sorts of metrics from it. They can also allow us to do group level analyses or other sorts of statistical analyses. So this is an example. These are real data and not really published, but these are real data. They kind of have them, I do have them published, but supplemental information. They weren't a primary aim of a study. But what this is showing is this is just showing group differences in cortical thickness in different parts of the brain. Meaning, if you measure how thick the gray matter is, you start on the outside, you go down to where the white matter starts, and again, there's a lecture about the anatomy. Encourage you to watch if you haven't watched that yet. But this is showing where there are these group differences, and what the groups in this case are doesn't matter. But what this is showing is these groups differ in thickness in many parts of the brain. This area in blue, light blue, is where this one group has thinner cortex, and orange, yellow, red is where that group has thicker cortex. Again, this is research questions that we can ask. Do these groups differ in cortical thickness? So do adolescents who have had early life surgery, do they differ in cortical thickness and again in adolescence than individuals who have not had surgery? That's not this sample, but that could be a question you could ask. Or is there a correlation between some behavior and the outer layer, the cortex of the brain? And we could get a picture like this. So we will do that in this class as well, is generate something like this. Different question, different data, but at least do the analysis. Then we have T2 images. So a T2 image is really useful for seeing brain pathology, or at least changes associated with pathology. So in here, white matter, which would be connections between cell bodies, is dark. And the outer layer, we have gray matter. It's also dark, but it's relatively lighter. So in here, white matter is darker, gray matter is slightly lighter in intensity. But then what we notice is the cerebral spinal fluid, which is inside and surrounding the brain, is bright. So this light fluid on T2 is very useful for seeing atrophy because we can see that contrast between water, essentially, the cerebral spinal fluid, and brain matter. And so we can look here. This is an area called the hippocampus. It's important for memory and learning and some other functions. And this individual has a little bit of extra space around there, this cerebral spinal fluid. The lateral ventricles here are larger than they would be probably when this person was younger. Same thing, this is what we call a third ventricle, and looking around the outside of the brain, there's just a little extra fluid. And so that's a sign that this person has some general atrophy in the brain. This is an older adult brain. This is not terribly abnormal for some older adult brains, mid-70s and later. But what can also show up on here is tumors can show up pretty clearly on T2 scans. And then this can also be useful for seeing some damage to the white matter itself. But in that case, our next sequence is going to be even more important. So this is what we call FLAIR, which stands for Fluid Attenuated Inversion Recovery. And this is a special type of a T2 scan. Again, don't worry right now, T1, T2, what those mean. Just those are terms, just understand what they look like. And so compared to a basic T2 like this T2 FLAIR, what it does is it subtracts out the signal for cerebral spinal fluid. And so now the lateral ventricles are dark, around is dark, but what we're seeing now is we're seeing signal within the white matter for the most part. Some of it can be within some gray matter structures, but within the white matter we see these areas of brighter, more intense white coloring. This is what we call a hyperintensity. And so in this individual, this happens to be, these white areas are where this individual has had small strokes that they don't know they're having. They just happen over time. If this was a different individual, it could be possibly multiple sclerosis. So that can produce lesions in the brain and be little changes, or these could be areas of edema, or again some sort of lesion. We don't always know what it is, but for older adults, which is the brain, this is a brain of an older adult, for the most part, these type of spots that we would see on this FLAIR image are going to be secondary to cerebral vascular disease, so they are small strokes. And it's relatively rare to see any sort of changes like this in young adults. Another consideration for imaging is what we call the strength of the magnet, the field strength. So a typical research scanner, such as ones we have at the McKnight Brain Institute, have a field strength of 3 Tesla. And that's about 30,000 times greater than the Earth's magnetic field. Clinical scanners are going to be 3 Tesla, 1.5 Tesla. And here's just an example. So 1.5 Tesla. The scanners, these are not always older scanners. They're still making, companies are still making new ones. They're just not quite as strong but they can be cheaper to buy, they can sometimes be cheaper to run, and sometimes they're faster to run. And again, but cost is a major factor, and also sometimes the 3 Tesla scanner, which I don't have an example on this image, but the typical this 3 Tesla scanner can sometimes be a little bit too strong for some individuals if they have some certain implants or whatever, and so you have to do the little bit weaker scanner. But then what this is showing actually is relative to a 7 Tesla MRI, which we do not have here at University of Florida. There is one at the Mayo Clinic in Jacksonville, and some other sites have 7 Tesla MRI. They can provide much more detailed images of the brain. There's a lot of challenges in running the scanners and dealing with the data, but as more time goes by, we're going to get more and more of these stronger magnets at different institutions for research. And some institutions have had them for quite a long time, but there's enough challenges with them that typically are sticking with 3 Tesla. So that all the images that we're going to deal with in this class are from a 3 Tesla scanner. So we'll get resolution. This is showing resolution possible, which you can get higher resolution with weaker scanners, in a large part by leaving people in the scanner for longer, which has its own challenges. But that's the difference. So field strength is going to be one thing that will influence the resolution of the image. Then here's just another example of 7 Tesla. But I want to highlight so we can see these are structures of the brain, but we can start to get a sense of some of the vasculature in there. And zoomed here into, this is what we call primary visual cortex. In the occipital lobe, you can see the different layers of the cortex. Most of the cortex has 6 distinct layers of cells. And with these scanners, we can see that. So this is a very high resolution image, very high quality, and again, can provide some really clear information for both research and clinical questions. Now we're going to talk a little bit about white matter of the brain. So this is what we call blunt dissection, where we can follow different white matter pathways. These are visual pathways going back to occipital cortex from part of the thalamus. This is corpus callosum, so these are connections between the 2 hemispheres of the brain. So if we want to look at the white matter of the brain, how do we do that? Again, if we're looking at these scans, what we notice is the white matter looks very homogeneous. Really what we want to understand are how do parts of the brain connect together? At least some of us do. So this is where diffusion MRI comes in. In this image, the mummers are holding hands. They're connecting as a network. We see this in social networks. You can see this on Instagram or Facebook or whatever kind of social media you belong to. You can be connected to somebody else. That person can be connected to somebody else, and on and on. But this is what diffusion MRI helps us understand, is how brain regions of the brain are connected together. And they could have direct connections, or they could connect through an intermediate area. So for example, over here we have visual pathway. So this is our optic tract going back. So the eyes would be here and get the nerves going out. And so the eyes connect back to an area of the brain called the thalamus. And then from there connects back to the occipital cortex. So eyes to the thalamus and then occipital cortex. And so really we have this intermediary structure, that's this thalamus. But that's what diffusion MRI can help us, is really understand how different areas connect together. And we can build up these networks. So as I showed already, here's a brain, gray matter, white matter. And it's a little bit like a cream-filled donut, where you have the outer layer, the white matter is kind of a nice gooey middle part. But of course, it's really not like that because we have structure to it. So using diffusion imaging, we can understand the gross structure and integrity of white matter pathways. So this is one of the tools we'll get some experience with, a tool called Tracula. We'll come back to that later in the semester. But we can understand some of the pathways in the brain, how areas of the brain connect to each other. And we can use this to make pretty pictures. It's part of what I do. This is a part of the brain that was important for my doctoral dissertation. So I made a nice picture from it. Again, we can take these images and see, this is what we call a track density image. And we can see areas in the brain where there's a relatively higher density of white matter fibers, which is what we're seeing here, and use this to see some really fine and small white matter pathways. And this is actually what we call a super resolution image, where if you've watched any of the, I don't know, like CSI shows and they enhance a photograph where you have the blurry face and it comes into clear picture, that's actually a little bit of what we're doing here with this image, where the resolution was much lower, but you can use sort of within, we'll come back to this term, but a voxel. You can use a little bit of information from inside this little space chunk of the brain that we're imaging to get a higher resolution image. It's a little artificial, but the process is the same of some of those enhancing images that occur. And then again, of course, we can look at connections between specific areas of the brain. We can calculate the nature and the strength of these connections and use that to test specific hypotheses about specific behaviors. We can even build up whole networks of the brain. So this is the whole brain, looking at a lot of different areas. How are they connected? How strong are those connections? We can use this information to run a lot of research questions. Diffusion-weighted imaging gets its start many years ago. And so really what we're doing is we're tracking movement of water molecules in the brain. And so they will move randomly in general, but sometimes in the brain there's some constraints on how they move. So the history of this goes back to the 1800s. Robert Brown was a Scottish botanist. What he did is he dropped pollen on the surface of water. As he watched closely, it moved around, even though there's no obvious reason for it to move. So it looked like the pollen was alive, but it wasn't. So what was going on, and what does this have to do with brain imaging? What Brown saw was diffusion. And as you hopefully remember from chemistry, diffusion is a physical process whereby a substance tends to spread steadily from an area of higher concentration to an area of lower concentration, resulting in a general equalization of concentration. So diffusion is a macroscopic manifestation of the microscopic principles of what it's called Brownian motion. So Brown described what he saw. So here's an example of what he saw. This is computer simulation. Random movement of pollen on top of the water. But then James Clerk Maxwell and Albert Einstein described the process mathematically. Einstein's description was published as part of what some people call his miracle year in 1905. Einstein published 4 groundbreaking papers, one on the photoelectric effect, one describing the relationship between energy and matter, E equals mc squared, one describing special relativity, and one describing Brownian motion. So his other ones, special relativity, energy, matter, E equals mc squared, those are the ones that get more attention, but that same year Einstein published a mathematic description of this type of motion. And so years after Brown, Einstein, and Maxwell had described this motion and the mathematics of it, the mathematics of Brownian motion or diffusion was used to help researchers follow water movement in the brain. And so we can differentiate between tissues based on how that random motion of water occurs. Gray matter, cerebrospinal fluid, there's relatively less tissue substance that is going to impede the diffusion of the water. And so we get movement that's relatively unrestricted, what we call isotropic. In the white matter of the brain, these are axons of the neurons. They can be bundled together. They can be wrapped in myelin that provides some insulation. This will do a relative restriction of diffusion in what we call a anisotropic diffusion. And this can be modeled in 3 dimensions, more isotropic, anisotropic, again, a more directional type of movement. So MRI can provide, you can run it and run what we call a diffusion sequence, but it can provide a diffusion encoding. And really what that means is we can apply magnetic field strength. We can vary it by what we call gradients, and we do that in different directions in the brain. And we'll acquire some baseline image where there's no diffusion encoding. It's kind of like a T2 image, but then we can acquire diffusion encoding, different directions. Again, don't worry about the physics of this for this class. Just the general principle is we get different directions, and then what that will do is that will allow us to understand how water moves in the brain, the directionality of movement. There's an example, varying gradient direction. We can see how intensity differs in different parts. So dark here, this corpus callosum, light, kind of midpoint. And again, what this is just showing is different directionality of white matter fibers in areas of the brain. We're going to do a short detour to introduce a term. The term I brought up briefly before, but what we have is, if we think about a picture, so you have your phone, you take a picture, and if you zoom in, you can see the pixels. So a pixel is a 2-dimensional chunk of a digital image that has some intensity value encoded. It can also have color and some other information. So in imaging the brain, because it's a 3D physical object, we want to have a 3D digital representation of it. This can be true for a lot of other imaging as well, generally photographs 2D, but I think going forward, trying to move the different creators and companies are trying to move forward to 3D, more 3D imaging. And so what we have is, in the brain really though, we're getting a 3D representation of the brain. So instead of pixels, we have volumetric pixels, which we shorten to voxels. So you see these in Minecraft, in a lot of different games and other things. We'll use things like a voxel to make 3D objects. And they can be different sizes, resulting in different resolutions. So again, Minecraft, you have big blocks, but we could also do a higher resolution, much smaller blocks. And they can be different shapes, but they are typically some form of a cuboid. And within research imaging, we typically prefer squares, these cubes. Again, we can look at that. So that's a voxel, that's the term. And so here we're showing a typical, for a research scan, a typical voxel size, the dimensions for diffusion MRI. It's 2 by 2 by 2 millimeters. So each side is 2 millimeters, which gives us a volume of 8 cubic millimeters. And what that will do is give us a picture of the brain, but it's relatively low resolution. There can be thousands and thousands of axons within that space, or thousands to millions of cell bodies and all sorts of different structures. So really we talk about microstructure, but this isn't microbiology. This is not cellular biology. We're at a higher level than that, lower resolution. So, but every voxel, going back to diffusion, we want to know, is this white matter, is this gray matter, is this cerebrospinal fluid? If it is white matter, what pathway is it part of? So what's the orientation of diffusion? What's the magnitude of diffusion? So where is the water going? How quickly is it going in a given time? And a grayscale image really can't capture all of that. So we get images like this where we can understand the dispersion of diffusion. And we'll get to a little bit of the mathematics of it. But again, the goal of diffusion imaging really is to understand the structure of white matter of the brain. And a lot of other uses clinically, but for our purpose, understanding white matter. So then there's a term called the diffusion tensor, or diffusion tensor imaging. So what we can do is we can use information. We have directions, X, Y, Z, and we can characterize the displacement of water in this three-dimensional space by looking at, again, this X, Y, Z plane coordinates. And diffusion in the brain, in some parts, it's relatively isotropic, meaning displacement water goes the same in all directions, but especially in white matter, that's not the case. It tends to go in one direction. And so we can use that information to get this tensor, use it what we call a tensor model. So we can express the notation of direction as a tensor. This is just one way. There are a lot other ways to do this, and this is what we call DTI, diffusion tensor imaging. This was essentially the first way of doing this. More recently, there are more complex models that are a lot better. But for simplicity, this is still one of the prevailing ways of understanding diffusion imaging. So a tensor is a 3x3 symmetric positive definite matrix. So it only has 6 unique elements. You have the diagonal, off diagonal, and again it's symmetric, so these are repeat. Some of you might have had linear algebra and you've dealt with matrices or other sorts of classes, you've dealt with these. If you haven't, don't worry about it. This is a math class, this isn't a physics class. This is just a little bit of background. So this is a 3x3 matrix, 6 unique elements. So really all we need to do is just estimate this upper or the lower triangular part. We just need 6. And we can use that to characterize displacement of water in the brain in 3 dimensions. And I say the brain, but you use diffusion imaging, we're looking at the body or all sorts of other things, but focusing on brain imaging, neuroimaging, so that's why I talk about the brain. So here's an example. So diffusion MRI, if you had a value of 8, sort of hypothetical, on our axis, 0 everywhere else, that means this is a perfectly spherical displacement. So this is physics, this is your spherical cow, meaning this is your underlying assumption of perfectly isotropic diffusion. It can be different rates depending on where it is, or there's other factors that can influence it, but here's how it would be. So the directionality, spherical diffusion, same in every direction, but the magnitude is smaller in B relative to A. But now if we have different numbers, what we're seeing is there's a primary direction here. So 633, diffusion in one direction is twice the size of the other directions, or 4 times, or 10 times, or we can get some sort of random, some other orientation of it. And again, so that's really what we're trying to characterize using with diffusion tensor imaging. Is water displacement spherical, or is it more shaped like a pencil? And that's really the goal of diffusion MRI. And these are all mathematical models, but they are biologically meaningful. A little discussion about eigenvectors and eigenvalues. So the tensor matrix and the ellipsoid can be described by the size of the principal axes, those are your eigenvalues, and the direction of the principal axes, the eigenvectors. These are represented by lambda 1, 2, and 3. We'll get into this. A tensor matrix. Lambda 1, 2, and 3 are termed the diagonal values of the tensor. Lambda 1 indicates the value of maximum diffusivity or primary eigenvalue. This is a longitudinal diffusivity. So that is identified first. Lambda 2 and 3 are then identified. They represent the magnitude of diffusion on a plane transverse to the primary one, and they are also linked to eigenvalues that are orthogonal to the primary one. Technical, again, don't worry about it. All it's saying, what this means is lambda 1 is identified first, so we have an eigenvalue and we can get an eigenvector. So magnitude of diffusion in the primary direction, secondary and tertiary, they all could be the same, but typically within the brain, within real biology, there's a little differentiation between them. And so lambda 1 is identified first, and then you have this is the primary direction. So you get a value and a vector. And then lambda 2 is defined, you get a vector that is orthogonal to the first one, meaning 90-degree angle. And then lambda 3 is identified, and that's going to have a vector that is orthogonal to both lambda 1 and 2. That's a little arbitrary. We won't get into how that can be an issue sometimes. And this is just background. But physical interpretation. So eigenvectors express diffusion direction. The values express the magnitude. So if 1, 2, and 3 are about the same, we would have a roughly spherical, this ellipsoid is going to be roughly spherical, and that means that we can think of this ellipsoid as the likelihood of water molecule displacement at that voxel, meaning how is it going within each voxel of the brain? Is it going relatively in all directions the same, which we would expect in cerebrospinal fluid? Right in the middle in those lateral ventricles or around the brain, that water, there's really nothing, not much to impede diffusion. So it could be spherical, but if we're in the gray matter, there's a little bit of restriction. If we're in the white matter, there's more restriction. And so we're going to tend to have a primary direction. So what this can look like in the brain, we can use this. So here's a T1 image. We have an intensity value at each voxel, but again, this doesn't tell us anything about the structure of the white matter. But if we get a diffusion image, we can overlay and look at that same voxel, and there's going to be some orientation, meaning water tends to go in a particular direction, a particular speed, if you will. And so we can get that for every voxel of the brain, really start to see some structure here. If we connected all these lines, we could start to track where these fibers are going. And we can color code it. We can say, in this example, red is primarily left and right movement. Again, going back left and right. Green is going to be anterior, posterior. Again, running front to back in the brain. And blue is primarily going superior and inferior, which is right here. And so now we can see there's this whole, potentially a whole white matter pathway right here. Here's a white matter pathway, and then if we're looking again white matter right here, these could be pathways, and those are biologically meaningful structures in the brain of white matter. So from this we can calculate some indices of diffusion. So these are metrics that we can use for research, and in this class we are going to calculate these. It's all automatic. We don't have to do anything by hand. We do our input of the images, and we have software that could spit these values out. But the simplest method is what we call mean diffusivity, and that is just the mean of the three eigenvalues, these lambda 1, 2, and 3. So equivalent to this orientationally averaged mean diffusivity. Again, don't worry about some of these terms like orientationally averaged, but that's just on average how fast is water going in that part of the brain is one way to think about it. This is an inverse measure of the membrane density, meaning in cerebrospinal fluid, out in the fluid, in the water. There's really nothing restricting movement, so we get a high intensity that's brighter because water's going as far as it can go in this given time. There's nothing impeding its movement. But once you start to get to the boundaries of the ventricles and out in the brain, then we get some impeding of movement. So we really end up with a similar mean diffusivity for gray matter and white matter. They're not that different. CSF is going to be higher. Again, water can go fast as it can in all directions. This can be sensitive to cellularity, edema, and necrosis, meaning that we can see areas that might show up relatively brighter on this type of mean diffusivity image. If we had a relatively brighter spot right here, that might be an area of swelling or something else has changed. It's different. And so that has clinical utility. Another metric is what we call fractional anisotropy, or FA. Here's the equation for it. Memorize it. It'll be on the final. Just kidding, we don't have a final. No tests. You don't need to memorize this, but this is the calculation of a metric. And don't worry about this. You can calculate this by hand for every voxel, every speck in the brain. I've done it for a few. But again, we have software that'll do this automatically. But this will provide a value between 0 and 1. This is helpful because this will tell us what the shape of diffusion looks like. So again, going back here, so A and B would both be 0. They're perfectly spherical. E, or an F, and D, but E and F are going to be very close to 1. D would be a little bit lower. So we're getting some value between 0, which would be spherical, all the way up to about 1. We don't ever quite get to 1 with diffusion MRI, although sometimes with the diffusion tensor calculation, because of some noise in the brain, other issues, we can get values higher than 1. Those aren't real values, and we tend to either get rid of them or just call them all 1. Anyway, what this is doing is it's going to provide information about the shape of this ellipsoid. So a higher FA value, closer to 1, is more pencil-shaped. 0, spherical. And we can use this information to do what we call it. We can also colorize it, and this will demonstrate the diffusion, the direction of the diffusion, the direction of the fibers, really. And so this is really meaningful. Again, red, left and right movement, green, anterior and posterior, blue, superior and inferior. So we can look at these different slices and start to understand structure of white matter of the brain. So this is, again, a colorized fractional anisotropy, a colorized FA image. Other summary measures, axial diffusivity, that's the greatest of the 3 eigenvalues. So in this case, that's just lambda 1. We can have radial diffusivity, which is the mean of lambda 2 and 3. Some people assume this radial diffusivity can tell us about the integrity of myelin. If we're looking at this in white matter, we can say, well, if we have higher radial diffusivity, then maybe there's a breakdown in myelin, this insulation that can impede the diffusion of water to some extent. It's more complex than that, but it's a relatively safe assumption a lot of the time. So these are other metrics that we will create in this semester. And these are all ones that can be looked at for research, to a lesser extent clinically. So there, as I mentioned earlier, diffusion tensor imaging is just one way to analyze what we call diffusion weighted imaging. So a tensor is an imperfect model. And really we're assuming there's one major, or this primary diffusion direction in a voxel. But what if there's more than that? This is where what we call high angular resolution diffusion imaging, or HARDI, can come in. So we can have more complex models. So here's a diffusion tensor, ellipsoid. We say, okay, there is white matter going that way something's going on. It's not quite pencil shaped. A little bit more complex, but that's what we see. Complex, we actually have some crossing fibers. Again, this is just a mathematical representation of the diffusion, but here then we can get this sense of we have these crossing fibers. What about this one? Crossing fibers, but the angle is not quite as intense. And then in some places, really just one primary direction of fibers, and we kind of get the same story. This is important though, because about 90% of white matter in the brain contains complex fibers, meaning it's not just a unitary direction. But diffusion tensor imaging boils it down to one number essentially for each voxel in the brain, which is very useful for a lot of research and clinical tools, but it's an oversimplification. So a lot of times we're using more complex models. We won't get a lot of experience with that in this class, just because that's we want to get the foundation for all of you. So to do a diffusion analysis, if we want to do a tensor, we want to remember this tensor has 6 unique parameters, so we need to acquire at least 6 diffusion-weighted images. And if we want to do this Hardy method, which is allowing for a more complex representation of diffusion, we need to typically acquire at least 30 different diffusion directions. And what you're doing is you're taking a picture of the brain, and then you take a picture of the brain again with this gradient applied in a different direction, so you're getting kind of more sensitivity to water movement in different directions. If we get enough of that, then we can acquire more complex models. But we only really, for diffusion tensor imaging, we really only need 6, but you do need about 20 to 21 is going to be ideal to have a more stable understanding of water motion. And for research purposes, we're typically acquiring more than 40. We can use this information to do what I hinted at earlier, tractography. So this is tracking fibers. And so this is really, it's not a measure of individual axons. These are mathematic representations of diffusion. So this is really big bundles of axons, if you will. And we can use fiber tracking. We can connect these voxels based upon similarities in maximum diffusion direction. So again, we can kind of draw lines and say it's going like this, and then we'll have some sort of rule saying if the angle is more than this many degrees, we're not going to go there because that's biologically unrealistic. And what that angle is, there's not a general consensus, but there are some typical ones kind of in the range of about 45 to 60 degrees. But that's the basic of fiber tracking. You can do it different ways. Won't get into all of this, but what I only want to point out is we can do what we call deterministic, we'll have more on the next slide, or a probabilistic tractography. And the probabilistic is going to be more powerful because, again, we're running sort of simulations of, well, what if it connects here? And then you get these probabilities that it's really can connect that way. And it really can allow for more powerful understanding of white matter. So streamline, this is deterministic. We're just going to connect the voxels to each other in what makes the most sense. And again, you're going to keep doing that until you get to some termination criteria. The angle is too sharp of a turn, or fractional anisotropy value drops too low, which in that case means we're probably out of white matter and there's nowhere else to go. You can do the probabilistic, as I said. You can get a final picture, so this deterministic type of picture, some fibers. After you do probabilistic, you can say, I'm going to use probabilistic, and then I'm going to take the most probable path or paths, and then we're going to draw these pathways. But a lot of times it's just kept a little bit more just probabilistic. Again, we don't need to worry about what all of this means. This is, unless you're going to go and focus on this more for a career, but this is just in the background, some of the tools we're going to use. As I showed earlier, this can be important because we have complex fibers, and we can have the crossing fibers, we can have what we call kissing fibers, and if we're using diffusion tensor imaging, we can end up with a low FA within the voxels. So going back to here, this is an image based off of the fractional anisotropy, and that's relatively low because remember, a higher FA closer to 1, that means more directional, so this is going to be higher, this is lower. But the problem is that this is a lower FA value, but it's not because the underlying white matter, like there's really no shape to it, it's just we have two very clear crossing fibers, and so they're kind of canceling the signal out. And that's where the probabilistic tractography can come in, or the more complex models. But let me show you some other comparisons between diffusion, the DTI, and Hardy methods. So here's an area toward the front of the brain. So anterior is up toward the top of this. This is an area we call the frontal forceps. And that's just an area where we get a lot of white matter that is connecting sort of front to back. We're getting area connecting left and right, meaning between the hemispheres, and also up and down. So it's a very complex area of the brain. But what happens on our FA images, so here's an image that's not colorized. Each intensity is just that value between 0 and 1. So closer to 1 is going to be brighter. Lower values can be gray matter or the cerebrospinal fluid. Those are closer to 0. And so we have this area in the white matter that's darker, zoomed in right here. So that means lower FA values, but it's not because the white matter is in bad shape. That's kind of what we assume. Low FA in the white matter, something's going on. There could be some damage. There was a stroke. Something is wrong with the white matter there, but that's an assumption that's not always true. And this is an example of that. We can have low FA because we have crossing fibers there. And what this ends up with is, let me show you this one. If we use this Hardy method, a high angular resolution diffusion imaging, we can see those crosses. This is corpus callosum. We get some white matter going that way, but we also get some crossing out and branching out, and so then that is the complexity of white matter. So sometimes we can get values, we could get changes. So hypothetical. Let's say we had a group of individuals, and we're interested in this part of the frontal lobes. We have a group of individuals who have selective loss of white matter connections that are going, in this case, let's say left and right, through this area. We could end up with higher FA values, these fractional anisotropy, not because the white matter is in better shape. Again, that's our typical assumption, higher FA is good, but we could end up with higher FA because we've lost the complexity of fibers in that area, and we want the complexity. Or maybe they have a group of individuals who are developing complexity in an area, and you don't want the complexity. But this is a discussion for another class, another day. We've been spending a lot of years working with improved methods, at least colleagues of mine have. So again, zooming in, complexity of white matter. Here's one other example, real data. So this is a diffusion tensor based tracking. So this is in the brain stem. If you don't know neuroanatomy, that's okay. Don't worry about it. Do have the intro to neuroanatomy, but this is brain stem. And so we have this blue pathway. This is what we call cortical spinal tract. It's going to include some other pathways, but this is motor pathways. This is primary motor cortex. This is your cortex, your brain, being able to tell your body how to move. And with DTI, what happens is we have a little bit of loss of understanding of this pathway as we're going through an area that we call the pons. And so that's an area where we have a lot of connections between the two hemispheres. This is cerebellum. You're just going to have to imagine this if you know it enough. But this is connections between the hemispheres, or connections out of one to little, what we call nuclei within the brainstem, and then to other parts of the brain. But we have basically, so this is an intersection. So we have a major highway or road going this way and a major highway going that way. And so now we have an intersection. In diffusion tensor imaging, we lose some of that signal. Again, we have it thinking, oh, now we have this information, this connection going, turning and going up. Is that real? Probably not. So using a improved method that allows for more complex modeling, we now see, oh, we can see that intersection. And here again, there's a little bit of suggestion of, we're picturing these are roads and cars driving on these. These are cars that are skipping the intersection and going. But that's really not what's happening. Is there is an intersection and they're just going through one another. Well, that's an example of more complex modeling and what it can tell us. But diffusion imaging is affected by several factors of the scanner and properties of the body. We end up with, and this is true for all MRI, but the diffusion imaging in particular, also functional imaging, is going to be affected by this. So we can get field inhomogeneities, we can get signal loss. These can be imperfections of the main magnetic field. It can be something about the person themselves. Maybe you have, you have sinuses up forehead, and maybe sinuses, you get an infection, and that can change the signal in the front part of the brain. So right here, this is the front part of somebody's brain, but that is not what the brain is shaped like. We don't have this big concave pattern. It's going to be more convex. And so that's distortion. That's not real. And that can cause problems for research. And there are ways to fix that distortion if we have the right type of images. Anyway, we're going to skip most of this, other than I want to introduce this term called eddy currents. So if you've watched water going down a river, especially if it's a little bit faster flowing, and you have, say, a rock or a branch or a log or something in there that's impeding a little bit of the flow, what you can get is these eddy currents, or over at the side of them sometimes, or this little counter current. And it can just sort of swirl around and maybe draw a little bit on this. So again, if you have a river flowing down, that's not letting me draw. Oh, there we go. So we have water flowing. If we had a rock here, again, what happens is we can kind of get this little swirling of the water as it's flowing down. Sorry for all the jumping around. But that's an eddy current. And so what happens with diffusion imaging is you're applying these gradients, meaning we're looking at water movement in different directions in the brain, and we're applying gradients. So we're think about it as lining them up different directions, or take them out of alignment in different directions. And what that does is that can introduce what we call eddy currents. And those can distort the image. So we have software that will correct for that. So to analyze diffusion data, we want to register. So this is more like a photograph of the brain, but over time. So it really is like, I say it's a static picture of the brain, but it's kind of like a video recording, series of stop of pictures, which is what a video recording is. But we use that information to get just one static picture of the brain. But what that means is we're scanning someone over time, and this is true for all imaging. So if you move your head, it's going to blur the image a little bit. So we can, and then with diffusion imaging, we're requiring multiple directions of information. And so really what we do is we pick one of these images, typically one without any diffusion, what we call a diffusion gradient, and we line all of the other images. We register them all to it. We're going to do this in the class. You'll understand what this means more. And then we can fit some sort of model. That can be a diffusion tensor. It can be something more complex. These are just terms for different methods. But we can use that to understand how is water moving in each voxel of the brain. Then we can do tractography, or we can do some sort of other metric. We can calculate fractional anisotropy, or whatever we're going to do. We can just calculate these metrics. Here's an example of both a script, or part of a script, and analysis. So I put this together for my dissertation just as an overview of my processing pipeline, input data, different tools. These are going to be tools that we will use. This free surfer we'll use, DICOM Nifty we will use, Eddy Correct. Some of these are ones we're going to use. And so each of these blue clouds, the different software, some of them are just subsets of other software. And then the gray would be different files and then output. But what I had done is I wrote a script to automate all of this. So I could just process, get everything I need, and be done without having to do it each step. So that's going to be a little bit of what we're going to do in this class. Is just that's why we're going to focus on a little bit of scripting. So what that can do is benefit reproducibility. And if we have 200 MRI, we don't want to do each of these steps one by one. That would take forever. So you can have the computer do it. It can run until it's done. So that's why, in part, we do scripting. Those are structural methods: T1, T2, flare, and diffusion. Now we're going to briefly cover functional methods. Videos you can watch. We won't watch them, but again, this is a dynamic view of the brain, and we can track activity by tracking oxygen or glucose metabolism, blood flow, electrical activity, and the assumption is that this is reflecting actual sort of brain activity, and hopefully we can link some behavior to this activity of the brain. So fMRI, functional magnetic resonance imaging, this is more of the video of the brain, and we can use this to link if we're doing a cognitive task, we can link activity of the brain to that task, or we can also look at how does your brain look, how does it look when you're at rest, when you're not really doing much other than being the scanner. So fMRI was developed in the 1990s. It's been informative for many populations. It allows us to assess patterns of blood oxygenation in the brain, and we can use this information to look at networks of connections. Areas in orange of this image are parts of what we call the default mode network. It's a major core circuit in the brain, meaning these are areas of the brain right here that when you're not doing anything are what we would call connected functionally, meaning we have blood flowing into this area of the brain, that area of the brain, these areas of the brain. There's some other ones little in there as well, but they're flowing kind of in and out of there at about the same time. The flow of the blood is correlated in these areas. And you could do this. This is one individual, and we can do this for group level and kind of get an understanding of these different networks. Blue would be areas where there's what we call an anti-correlation, a negative correlation. So if blood is flowing in and out of this area, it's kind of suppressed, or vice versa. If it's elevated here, it's suppressing this area, if you will, one way to think about it. So the fMRI is based off of the BOLD signal, blood oxygen level dependent. This is really an indirect measure of neural activity. So our brain is working, neurotransmitters, something's going on, that will require an increase in blood oxygen. And so we get this oxygenated hemoglobin coming into an area for use, and then that's what we're picking up as the fMRI signal. Very brief introduction to it. There's other modalities. We don't cover them in this class. And we only cover what we call resting state fMRI. We'll come back to this later. But again, that's an indirect measure of brain activity. Almost done. Common MRI metrics. So structurally, we get volume of structures, thickness, that fractional anisotropy, mean diffusivity. We can get a shape of a structure. We can do connectivity, all sorts of different metrics. Functionally, we tend to do connectivity, or it might be the number of voxels that are correlated with activity on one part, but some sort of what we call a connectivity metric. We can get a similar type of metric with structural imaging. So example of volume, again, structure, it takes up space. And we can end up with, let me get this video going, as I'm scrolling through, we can use, this would be a tool that we'll use coming up pretty soon. We can get volumes of the white matter in green and white, lateral ventricles, the thalamus, putamen, globus pallidus, all of these different structures. We can get volumes of these structures. How many cubic millimeters or cubic centimeters are they? And that can be meaningful from a clinical standpoint, but more importantly for a research standpoint. And different views of the brain, but we can get volumes of each of these structures. So here's an example of research. This is a study from some years ago. Relationships between years of education and gray matter volume in healthy older adults. There's an area right superior temporal gyrus that correlates with years of education. This isn't a really large study, so take this. These findings are more preliminary. Here's another one, anterior cingulate gyrus, positive relationship between volume and the years of education. Of course, this is a correlation, single time point, so we don't know which came first. More education increases volume in these areas, increased volume associated with these people who go on and get more education, whatever it is, a little bit of bidirectional is probably, could be reality. And then again, another one, years of education, volume here. And this is a type of analysis we'll get some experience with, but again, this is volume and some behavior. And so your interest could be that, years of education, maybe your interest is performance on a memory task, or cannabis use, or whatever behavior. We can ask questions and look at relationships between volume and some metric. We can also look at volume of changes in the white matter. So this was a paper we published 10 years ago in the journal Neurology. We showed that within dementia, so these were older adults who had dementia, once we had more of this white matter disease, this term LA, it stands for leukoariosis. These are just those bright spots that are showing up on the scanner. You can see them here. These are areas that are generally stroke. And so if you have people who have dementia, who have a lot of evidence of these stroke areas, then they end up having more problems with what we call executive functions, being able to plan and organize, have mental flexibility. And so if you have dementia and have a lot of this, what we call white matter disease, then more likely to have some of those challenges, as I said, in mental flexibility and organization and planning and how quickly you can do things. And what it takes, and where do we see that tipping point? So this paper, we showed that the tipping point was actually here at about 3%. So if you took the whole volume of white matter and the volume of this white matter disease, once you have about 3% of your white matter volume affected by these strokes or these changes to the white matter, then that was going to be where somebody would start to develop, be more likely to have what we call these executive dysfunction. problems with the planning and organization. Again, this is only within dementia. But we do have some unpublished data suggesting that we see some similar patterns within sort of what we'd otherwise call normal aging. But this was kind of interesting finding, because this was a level that a lot of radiologists, neurologists wouldn't even comment. They would say, oh, there's a little bit of damage, and then it would focus more on, say, just these pure memory problems that somebody might have. What we're showing is this is going to be meaningful for clinical presentation and management of symptoms and all of this. Anyway, so that's based off of volume. We can also look at the thickness of different structures. Typically, this is going to be the cortex. And so here's an example. This is a publication from 2016, and the researchers said if you're looking at the cortex of the brain, said we really want to use thickness rather than volume. I mean, you could get the volume of the outer layer of the brain, the cortex, and it can be okay to look at that, but the preference is to do a thickness measurement, and that's just how many millimeters from the outer surface of the brain down to the white matter. And the volume's important, but for some analyses, at least consider thickness. So how does thickness look like in a research setting? So I showed a picture earlier with the blue areas and the yellow-red areas, this group difference in thickness. So here's another one, not one of my studies. So this was looking at thickness in what we call mild cognitive impairment relative to older adults who don't have cognitive impairment, or Alzheimer's disease versus older adults without Alzheimer's disease, and can kind of see the differences where there's a difference. And we're also looking at changes over time. And what we can see, so these are areas of the brain where there's thinner cortex and also increased rate of thinning over time, so more atrophy. So we have a lot of temporal lobe, part of the brain we call the insula, a lot of parietal lobe, some frontal. Alzheimer's disease, we can see a little bit more extensive. So the brain is it's thinner in these diseases, but then it also is atrophying, so it's changing over time faster. You're losing more and more of the brain faster and faster. And so these are areas that are, again, so that's thickness, and just one example of how we can use thickness measurements that we will get using software. This semester you can do that on enough people, a sample, and do studies like this. We can also look at the shape of structures. Other groups have looked at, so these are subcortical structures, we call it. So there's caudate nucleus, thalamus, putamen, we have amygdala, hippocampus, there's a globus pallidus in there. Anyway, these are different structures, gray matter, structures within the brain. And so in this one, this research team was interested in effect of HIV infection and age on subcortical shape, and showing that there were these relationships that you could see. And this is within these structures. So if we just did the volume of a structure, we might not see specific effects. So this is really like local effects, that certain parts of structures seem to be more affected by HIV than not just the whole structure itself necessarily. A lot of the thalamus is, but only parts, say here, of this caudate nucleus are affected. And so that's a similar one that I had done some years ago with, published with Parkinson's disease. What we had shown is, so one of the big questions with Parkinson's disease is, do we see changes on one side of the brain more than the other? Part of this is because of Parkinson's disease, let's say there is a tremor. That's just one of the symptoms. You don't even have to have a tremor to have Parkinson's disease, but let's say there's a tremor. The tremor tends to start on one side. So let's say it started on the left side. Do we see changes in, so the left side of the body, if it's starting tremor in the left hand, that's the right side of the brain. So would we see more changes within the right hemisphere of the brain than we would in the left hemisphere of the brain if someone has left-sided symptoms predominantly? And so that's a little bit of the background to this, but using this shape analysis to show, is there a difference within subcortical structures? And so what we're showing is, even though we didn't necessarily see volume differences, we did see some shape differences. So the caudate nucleus affected, putamen were affected, and these are structures that we would expect to be involved. They are involved in movement. They are affected in Parkinson's disease, but we're able to see that relatively early in Parkinson's disease, we can see changes in the shape of these structures. But a little bit more importantly, what we were able to show is individuals who had left onset Parkinson's disease, that means left hemisphere, we would expect a little bit more on the right side of the brain. We saw that relatively a little bit more affected right caudate nucleus, right putamen than the left side. And so that was kind of matching this hypothesis. And these are data that really hadn't been shown before, this link between, again, left onset Parkinson's, a little bit more affected right caudate and putamen. It made sense theoretically, but hadn't been shown before clearly. In the right onset, not quite as clear of a story there. So there's something, meaning we have a little bit the caudate, we didn't. So right onset, we were expecting left side of the brain more affected, as we saw with the caudate nucleus, but not really with the putamen. One of the downsides of this is that these were individuals who were not only left side symptoms. This is just what side do they start on. And some had progressed to bilateral. We kind of controlled for some of that, but again, a little bit of a hint. Right side onset Parkinson's, at least for the caudate nucleus, that left caudate was affected, but the right caudate was not. Again, not different relative to older adults without Parkinson's disease. But that's a shape analysis, and we will do the underlying image processing that could allow you to do this. We won't get into the shape analysis itself, but again, this is a metric, a way to understand and analyze the brain. Thank you. That'll be it for our presentation today.